Proteins must have a very specific three-dimensional shape if they're going to function properly inside the cell. So proteins themselves are made of amino acids. Those amino acids get linked together so that we have this long molecule that we call a polypeptide. So the polypeptide is the amino acids joined together, but it itself doesn't have this defined three-dimensional shape. So there are many steps that have to take place to get that polypeptide to a very specific three-dimensional shape. And that three-dimensional shape can take a lot of different forms. Sometimes it might have kind of a U shape, like you see right here. Maybe it's more of what we would call a globular protein, where it would be something along these lines. Maybe it's going to be more like this, okay, where it's long and slender and it extends across a membrane or something along those lines. Whatever the shape is, there are a lot of steps that have to get it to that. So we have different levels of protein structure, and I want to very briefly introduce those different levels. Those levels are going to take us from the undefined polypeptide up to the functional protein, which is what this side is, that is properly folded. Okay, so let's take a look at those different levels of protein structure and talk about the main differences between them. Here's a look at the 4 levels of protein structure. I'm going to go through and emphasize what the major differences are amongst them and also the type of bonds that are holding them together. And those are the main things that you want to focus on. The primary structure is really what we've talked about already. The primary structure is going to be joining the amino acids together in the correct order. And the information for that is what the DNA does. So our DNA, we consider it to be the blueprint. Well, it is the blueprint for how our proteins are constructed. So all of these little beads that you see here in the primary structure, those are the amino acids. They've been joined together in the proper order by peptide bonds. Okay, so those will be the type of bonds that are involved, and of course dehydration reactions would be what put those together for us. The primary structure itself, that's just what we call the polypeptide. It does not actually have any defined shape, and it's certainly not going to be functional. Now once that's put together, the second step is going to be the secondary structure. And portions of the polypeptide will undergo secondary structure. There's 2 different types of secondary structure, and you can see that shown over here in the secondary structure image. And one of them is called a beta sheet. So this one would be the beta sheet. The beta sheet, if we were trying to look at the overall structure of that, it's going to have kind of folds in it. So if I draw that again right here, just getting rid of kind of the extra stuff, it's a folded area. The other portion or type of secondary structure is what we call the alpha helix. And the alpha helix is just like it sounds, it forms a helical structure. So some of our polypeptide will get folded in these defined pleats, and some of them are going to get folded into helical structures. And what holds together the secondary structure is going to be hydrogen bonding. So hydrogen bonding is the key to the secondary structure. That's something that you want to remember. And the other important part is that it's hydrogen bonding between parts of the backbone. And what we mean by the backbone is that these are parts of the polypeptide that are not side chains or not R groups. The R groups are going to get involved later on. Once we get the secondary structure together, there is a little bit of a shape to our polypeptide or to our overall protein, but it's not going to be functional yet because it's not all the way there to that perfect shape. If we move on and talk about the tertiary structure, the tertiary structure is the portion where we start to use the R groups. And remember, R groups is just another way to refer to the side chains. And so the R groups are going to be used, and they will interact with each other. And now when we say that they're going to interact with each other, how they interact with each other, the type of bonds, the type of interactions that are involved, there is going to be a lot of diversity there. And that's because the R groups themselves are very different from each other. Some of them are polar, some of them are charged, some of them are hydrophobic. So if we were to list the type of bonds or interactions that you can have with tertiary structure, there will certainly be covalent bonds there. There will be ionic bonds. We'll have some hydrogen bonds. We'll have some of what we call hydrophobic interactions, which is just where the hydrophobic groups like to group together with each other. Once we get to the tertiary structure and we get that completed, for a lot of proteins, that's as far as they'll go. So they will only have the first 3 levels, and they will then have their perfect shape, they're folded, they're going to be functional. In a few cases, there are going to be multiple polypeptides that need to individually fold, and then they need to assemble together. And that's what the quaternary structure is. The quaternary structure is going to be when multiple polypeptides fold up, and by fold up, we're talking about them going through the primary, secondary, and tertiary structure. So they're going to fold up and then assemble to make a larger structure. And that's what all the different chains are right here. They're different polypeptides that have assembled together. In order for the protein to function, they need to fulfill all the different levels. So primary, secondary, tertiary for most. Some of them go through all 4 of them. If any one of the different levels fails, then you're probably going to have a protein that's not folded correctly and it's not going to function properly. Now, one last thing to talk about here is what happens if the protein unfolds. So it is possible that sometimes we have this perfectly folded protein and then something changes and it causes it to now get all unfolded. When we have a protein that unfolds, that process is called denaturation. So denaturation is the process. Denaturing is what we would call the protein unfolding. Now, denaturing really means that the bonds holding it together, and holding the folding is what we're talking about. So the bonds holding the folding together are disrupted. And by disrupted, they are broken, they are no longer formed. So what can do that? What can cause the protein to just unfold the way that we're looking at it here? Well, changes in the conditions that the protein is subjected to. So this would be any changes in the physical environment could do that. So changes in the physical environment would be things like temperature changes. So if the temperature goes up, then maybe we're going to break hydrogen bonds, and that would disrupt the secondary structure and it would unfold. This could be changes in pressure could affect that as well. So that's another change in the physical environment. We could also have changes in the chemical environment. So if we're talking about changes in the chemical environment, the type of changes that we're talking about there might be changes in the pH. We could have changes in ions. So there may be an abundance of ions. Some of them may have gone away. We may have other proteins that are interacting with this and causing some problems. So just emphasizing here, this would be something that is functional when it's folded, but denaturation is going to cause it to be non-functional. So basically it's not going to work anymore if it does get unfolded like this. Occasionally, it is possible that if the changes down here, if they're reversed, that we can get back to this side. Sometimes we can do that, sometimes we can't, depends on the protein and the severity of all of it. If we can go back to the folded state, we call that renaturation.